You know, when we talk about Alzheimer's disease, we often think about things like diet, exercise, or the environment. But what if one of the biggest clues was already written inside of us, in our own DNA? Today, we're going to zoom in on one specific gene, a protein powerhouse called APOE, and find out how one faulty version of it can literally change everything.
Okay, let's just jump right in with a number that really tells the story. 15 times. Yeah, if you happen to inherit 2 copies of a particular version of this gene, it's called APOE4. Your risk of developing Alzheimer's skyrockets by up to 15 times. This is not some small statistical blip. It is the single biggest genetic risk factor we know of for late-onset Alzheimer's, period.
So how on earth does one little change in our genetic code lead to such a huge risk? All right, first up, let's meet the main character in this whole story, the apolipoprotein E gene, or just APOE for short. It's time to really understand what it is and why it's such a big deal. So, the APO gene holds the blueprint for a protein that is absolutely vital for a healthy brain. Think of this protein as a tiny shuttle bus. Its main job is to drive around cholesterol and other fats, or lipids, delivering them to our neurons to keep them running smoothly. But here's the twist. This gene isn't the same in everyone. It comes in 3 common flavors, or alleles. There's APOE2, which is actually protective. Having it is a good thing. There's APOE3, which is the most common and pretty much neutral. And then there's APOE4, the high-risk one we're focused on today. Now, you'd think the difference between the good version and the bad version would be massive, right? Get this. It all boils down to a single building block, one amino acid at one specific spot. The common APOE3 has an amino acid called cysteine. The high-risk APOE4 has arginine instead. That's it. But that one tiny swap makes the entire protein fold up into a completely different 3D shape. And you have to remember, in biology, shape is everything.
So now we have this protein that's all folded wrong. And that brings us to a really key concept. This new shape doesn't just work poorly. It actively causes harm. The protein goes rogue. Scientists call this a gain of toxic function, where it becomes a villain in the story, directly helping to damage the brain.
So what does it actually do? Well, a perfect example is how it messes with beta amyloid. That's the sticky stuff that creates those infamous plaques in Alzheimer's. Because of its weird shape, the APOE4 protein latches onto beta amyloid way more aggressively. It's like a magnet, supercharging the clumping process and helping build these destructive plaques all around our brain cells. And the damage doesn't stop there. It gets inside the cell too. See, inside our neurons, there's another protein called tau. Normally, tau is like the wooden ties on a railroad track. It keeps the internal highways of the cell stable so nutrients can get where they need to go. But APOE4 encourages a chemical change in tau that makes it go bad. This altered tau lets go of the tracks and starts clumping together into what we call tangles. And without those supports, the whole highway system inside the neuron just collapses, which eventually kills the cell from the inside out. So we've seen the damage APOE4 actively causes, but that's only half the story. The problem is a double whammy. Now we have to talk about its loss of protective function. This is all about the really important jobs that the healthy versions of the protein do, but the APOE4 version, well, it just completely fails at them. Think about your brain's garbage collection system. The healthy APOE2 and E3 proteins are the all-star cleanup crew. They're great at helping clear out all that toxic beta amyloid. APOE4, on the other hand, is awful at it. And it gets even worse. It actually gets in the way, competing for the same disposal routes that other cleanup mechanisms use. So it's not just that it isn't helping, it's creating a traffic jam that stops the whole waste management system from working properly.
So here's the big question, right? It sounds simple, but the answer is anything but. If your brain has this amazing, dedicated cleanup crew, how can one tiny little change in your genetic code cause such a massive failure? That is the mystery we're going to unpack today.
Okay, first things first. Before we can understand how a system breaks, we really need to get a handle on how it's supposed to work. So let's meet the hero of our story, the brain's cellular cleanup crew, a process known as autophagy.
I want you to think of autophagy as your cell's all-in-one recycling and garbage disposal service. It is constantly at work, collecting old, broken parts and all this toxic protein gunk to keep your cells, especially your brain cells that have to last a lifetime, healthy and running just like they should. Without it, trash just piles up, and the whole system would grind to a halt. So it all boils down to this really elegant 3-step process. First, the cell collects all its garbage into this special little container.
Then, and this is key, that container has to merge or fuse with the cell's recycling center, which is called the lysosome. And finally, once inside, powerful enzymes just dismantle everything, breaking it down so the parts can be reused. It's a beautiful and absolutely essential system.
All right, now it's time to introduce the villain of our story. And what's interesting is that it's not some foreign invader. Nope, it's a variation in our very own genetic code, a faulty little switch called APOE4. And here is why we are so laser-focused on this one gene. Carrying the APOE4 variant is the single biggest genetic risk factor for developing late-onset Alzheimer's. So for decades, scientists have been trying to figure out the big question: why? What is it actually doing? And this brings us to the absolute core of the issue. It looks like APOE4 doesn't really introduce a new poison into the brain. Instead, what it causes is a loss of function. It takes that elegant, essential cleanup system we just talked about, and it breaks it. It's an inside job. It sabotages the machinery. So how in the world does it do that? Well, the sabotage is a multi-pronged attack, and it starts right at the source by messing with the genetic blueprint itself. Okay, check this out. On the left, you see the normal process. Your DNA has these specific spots that act like on switches to create all the parts for the cleanup crew. But over on the right, you can see what happens with APOE4. The APOE4 protein can physically park itself right on those same switches, basically blocking them. It's literally like putting duct tape over a light switch. The instructions to build the cleanup machinery never even get sent out. Now, what's so powerful here is that this isn't just a theory. Researchers actually looked at brain tissue from Alzheimer's patients after they had passed. And this chart shows you exactly what they found. In people who had 2 copies of the APOE4 gene, the levels of those instruction molecules, the little messengers that carry the blueprints, were way, way lower. The evidence is right there in the data. But the sabotage doesn't just stop with the instruction manual. This is where it really becomes a perfect storm, because even for the few parts that do get made, APOE4 finds other ways to break the actual assembly line of the cleanup process. Hey, remember that super critical second step, the one where the garbage bag has to fuse with the recycling center? Well, APOE4 messes that up too. It's linked to higher cholesterol in the brain, which messes up the cell membranes and physically stops that fusion from happening. It creates this massive traffic jam, with bags of cellular trash just piling up, completely stranded with nowhere to go. And on top of that traffic jam, there's another huge problem. There's a protein called mTOR, which is like a master regulator. When it's on, the cell is in grow mode. When it's off, the cell switches to clean mode, which fires up autophagy. Well, in brains with APOE4, mTOR often gets stuck in the on position, and that acts like a system-wide emergency brake on the entire cleanup operation. And get this, the damage even extends beyond the neurons themselves. Our brains have these critical support cells called astrocytes that help with the cleanup. But in mouse models, astrocytes that have the APOE4 gene are also terrible at their job. They just can't clear out toxic proteins effectively. Now, there is a little glimmer of hope here. When they treated these mice with a drug that basically forces autophagy to turn on, it actually helped rescue some of that function. So, let's put it all together. You've got sabotaged blueprints, you've got a broken assembly line, and even the helper cells are impaired. When you combine all of that, the consequence is pretty much exactly what you'd expect: total system failure. And this is the devastating domino effect of that perfect storm. With the cleanup crew totally out of commission, the very toxic proteins it's supposed to clear, beta amyloid and tau, they just start to pile up. They form the plaques and tangles that are the classic hallmarks of Alzheimer's disease. The system is so broken that it not only lets the junk build up, it actively prevents itself from being able to clear it, accelerating the whole tragic process. And you know, that brings us to our final and, I think, most critical question. If APOE4's main crime isn't adding some new poison, but simply breaking an existing vital system, does that mean that if we could find the right tools to reactivate it, or maybe just bypass the broken parts, we could actually fix it? That is the profound and surprisingly hopeful question this research leaves us with.
And there's another crucial housekeeping job that it messes up: synaptic pruning. The brain support cells, called astrocytes, rely on the APOE protein to help trim away old, inefficient connections between neurons. This is totally normal and essential for learning, but the APOE4 variant gets in the way of this process. It's like having a gardener who can't prune the dead branches, leaving the brain's wiring all overgrown and inefficient. making it harder to form strong new connections.
You see, the damage from this one little faulty protein just keeps spreading out, like ripples in a pond. It's not just about the plaques and tangles we always hear about. So now, let's look at the deeper cellular damage and see just how far these ripple effects go.
What we're looking at here is basically a three-front assault on the core machinery of our brain cells. First off, APOE4 messes with autophagy, which is the cell's own internal recycling program for getting rid of junk. Second, it attacks our mitochondria, the little power plants in our cells, leading to a major brain energy crisis. And third, it helps cause insulin resistance in the brain, which screws up how our neurons use their main fuel, glucose. It is a total cascade of cellular chaos.
Okay, I know, that was a whole lot of bad news, and it's easy to feel a little hopeless. But this is where the story pivots. This is where we get to the hope. Because understanding the problem in this much detail, knowing exactly how APOE4 causes all this trouble, is finally paving the way for some incredible brand new solutions. This quote really sums up the challenge perfectly. Because APOE4 is a double-edged sword. It adds a toxic problem while taking away a protective function. You can't just create a simple drug. You can't just block it because the brain needs what it does right. But you can't boost it because that would just increase the toxic stuff. It's so tricky. And that's exactly why these new approaches are so brilliant.
So how are researchers getting around this problem? With some really clever strategies. First, there's gene therapy. The idea here is to just deliver the protective APOE2 gene directly into the brain, basically giving it a better set of instructions to work from. Then there's antibody therapy, which uses custom-built antibodies to hunt down and help clear out those toxic APOE4 and amyloid clumps. And maybe the most futuristic of all are small molecules. These are drugs designed to be structure correctors. They literally find the misfolded APOE4 protein and nudge it back toward a healthier, non-toxic shape. How amazing is that? Which brings us to this one final massive question. The story of APOE4 shows us how one tiny flaw, one molecular mistake, can trigger decades of destruction in the brain. But it also shows us an incredible new path forward. If we can create therapies that are precise enough to fix that one single faulty protein, could we actually prevent this entire devastating disease? That is the unbelievable promise that is driving this entire field of research forward.